So I guess we can move on now to Justin and Patrick. Um, and I guess just to introduce myself, because I didn't do that at the start, I am a PhD student at UC Davis, and my research is focused on developing scaffolding and biofabrication techniques for cultivated meat. And now Patrick and Justin are in a different lab at UC Davis, so we'll hear about some of their research. You guys want to take it away?
Yes, thank you, Nick. I'll start sharing my screen. Okay, can everybody see the presentation?
Yep.
Okay, cool, thank you. So I'm Patrick, and this is my lab partner, Justin. We're both PhD students in the McDonald Nandy lab, and we're going to be talking about cultured meat research in our lab. So I first want to give a brief overview of our lab because it's not traditionally about cultured meat. So this circle might give a good description. So the left brown side of the circle represents traditional work with plants, where plants are typically thought of as a food source, a way to treat water and air, aid our behavioral health, etc. But the right blue side is what our lab is specialized in, which is using plants as a molecular foundry for the production of a variety of useful recombinant protein products. And so the lab works mainly on cell culture reactor systems and whole plant systems, which is basically using the plant as a bioreactor. And looking at this table on the right-hand side, we believe that plants should be more widely used for protein production because they are economical, have high product quality, and low contamination risk, making them a well-rounded system, often better than other conventional production systems. And so a brief overview of how our lab works, our lab development strategy. When we start a project, everything is very unknown, and we put it through this kind of pipeline, which can be summarized in modeling a process, designing genes, building our genetically modified plant, and testing protein expression, which leads into scale up, and that feeds into our model again. And so using this workflow, we have successfully developed many recombinant protein production processes in both whole plants and plant suspension cultures and bioreactors. And we can apply this pipeline and the expertise in our lab to a totally different project, cultivated meat production. And so this is an outline of the project's, the cultivated meat goals in our lab. And so the first is a techno-economic analysis, or TEA, and sustainability analysis of the entire production process, basically modeling an entire cultivated meat facility. And the second is the production of recombinant gelatin for use as microcarriers for meat cell growth and differentiation. And then finally, production of rice cells as an edible microcarrier or scaffold. So I'll go into the first project and then also just give a brief overview of cultivated meat and why we need it. So the global meat demand is expected to double by 2050, but doubling conventional meat production is not feasible because we have limited land and resources. And so a potential solution is cultivated meat, which consists of taking a biopsy of stem cells from an animal and having those cells proliferate in a bioreactor, and furthermore inducing differentiation of those cells into mostly fat and muscle cells to replicate a meat product. And so obviously this requires no livestock, little land use, and much less emissions. So we can say with certainty that this will definitely be environmentally feasible compared to current meat production. But we have to ask ourselves, is this going to be economically feasible? And there's much uncertainty. But current studies and knowledge indicate that the price of this cultivated meat is likely to be an order of magnitude greater than conventional meat of about $4 to $5 per kilogram. And so the ultimate goal of this research is to reach price parity with conventional meat production, and a techno-economic analysis, or TEA, can help us reach this goal. And just to describe a TEA, that's the assessment of technical viability, economic viability, and environmental impact of a production process. And that can be visualized in this little figure here, in which any part of the process can be modeled. And so what do we currently know about this sort of work? Well, in the last few years, the industry has grown rapidly, over 40 companies internationally, and there's much financial investment. And most of these companies are very pressed to start producing. But even though they may be working on process design, it's confidential and there's little collaboration. And so we make a very good argument that this should be done in academia. But in academia, there hasn't really been a focus on this yet. most of the focus in literature has been on the science. But if such process design and modeling work were done in an academic setting, there would be a better facilitation of knowledge and more freedom to explore the best possible facility design. And pursuing this research, I'll go through and test multiple design choices, ultimately finding and modeling the best facility. And that will pinpoint technical bottlenecks that will be addressed to make the process more feasible. And so these are just some current struggles with TEA research. There's bioreactor design, modeling scaffolds, choosing an appropriate cell line, and then pricing media, which I will focus on. And so, you know, as you can see from these two pie charts, from these two existing TEAs, media is the main cost bottleneck, accounting for most of the cost of the facility. And so this indicates that we need better ways of producing media components cheaply, and it also highlights how we must be careful when modeling or approximating different components of the process. And now I'll dive into that a little bit more. So just to describe here, the one problem is accurate approximation of the price. And so we first have to approximate the unit price of a media component in dollars per gram. And so one option is looking at vendors, but this may vastly overestimate the pricing media components such as growth factors at millions of dollars per gram. And so an important thing is to take into account the demand of the component and the market. So this log-log correlation gives an accurate price per demand of the product. And so this takes into account economies of scale. So that's one aspect. Another aspect is determining an appropriate or accurate concentration of the component within the volume of the media. And then you also have to decide on the volume of media required for the facility, which is dependent on your facility design. So once you can accurately price your media, now you have to figure out what are ways you can reduce the price. And so we have a couple of ideas. First is having an in-house recombinant protein production process rather than having to rely on other companies to source your media for you. And our lab is well equipped to do this, as I've shown you with our work with recombinant production in plants. And then another idea is using plant hydrolysate, or basically plant biomass, as a supplement for amino acids in the media, and this can be done very cheaply. And so so to transition to my partner Justin, another specific part of the process that our lab can experimentally address is gelatin production for scaffolding of the meat cells.
All right, so as Patrick mentioned earlier, my work focuses on leveraging plant expression systems to source microcarriers. The first project that I will talk about is the production of gelatin in rice cells to be used in microcarrier production. As a quick overview, muscle cells, which are the most abundant cell type in many types of meat, are adherent by nature and require a surface to proliferate on. In culture meat production, this surface can be provided by microcarriers, which are essentially small beads of various compatible materials that cells will preferentially adhere to. One of the materials that is used to make microcarriers is gelatin, which is something that we often find in other food products such as Jell-O. However, gelatin typically comes from animal skin and bones, and with the entire idea of cellular agriculture, we're trying to decouple ourselves from any animal-derived products. Consequently, being able to source gelatin specifically in rice cell cultures with my work could be a potential solution to this problem. For rice cells to produce gelatin, first we had to determine that the COL1A1 gene here was responsible for our protein of interest. The native gene has a few components that we are able to remove as they encode it for functions that we would not need in our final product. As a result, we really only took this green region of the gene that codes for the alpha helical region, and we placed that into our expression cassette as part of our vector design. The expression cassette includes several additional genetic components that will ensure that our transgene is expressed properly in the rice cells, and it will also subsequently allow us to identify successfully transformed rice cells further down the line. So currently, we are synthesizing our plasmid that will be used to create our transgenic rice cell lines. once we have actually created these transgenic rice cell lines, there are a series of tests that we'll have to perform to verify that our early design decisions are working as intended. That I won't really have time to go over today. But assuming everything goes well, I wanted to quickly highlight that our lab has a unique capability to scale the production of these rice cells. So our lab actually has a 140-liter pilot-scale bioreactor where we will be able to closer approximate the realistic production scales that you see versus what you typically see in a laboratory, which is the 250 ml shake flask, and then perhaps these 5-liter benchtop bioreactors that we have in our lab as well. And here, I apologize for the quick transition, but the third project that we are working on is actually looking at rice cells as microcarriers. So in this, we are actually, rice cells in suspension cultures will grow as aggregates, and we may be able to use themselves as microcarriers. So these aggregates are in the size scale of several millimeters, and you can kind of see what they look like in these two images on the right-hand side. The aggregates themselves are approximately spherical in structure and are relatively porous as well, making them good potential candidates for microcarrier production. When compared to other traditional microcarriers, we've identified that these rice cells have several advantages. One of them being that since they're sterilely produced, there will be no additional processing before we can put them into the bioreactor. The rice cells themselves are edible and have a designation from regulatory agencies generally recognized as safe. So in the final product, we actually won't need to have to purify them out from the meat. And additionally, with the experience in our lab with recombinant protein production, we actually believe that we can program and tune these rice cells to express different proteins that at the end will actually support the growth of muscle cells better. So with that, we're really humbled that ProteinX invited us to talk a little bit about our research and kind of share what we've been doing. We also want to say just thank you to all our lab members who wouldn't be able to do this without them, and also to the Good Food Institute and the National Science Foundation for providing funding for Patrick and I. And then lastly, to the UC Davis Culture Meat Consortium for providing many opportunities for collaboration as well in this space. And that's it. Thank you.
Awesome. You guys are doing a lot of cool stuff. So I guess you gave the kind of the overview of what you're looking to do in the lab. Could you give just maybe a brief comment on where in that process you guys are at the moment?
Yeah, so I can kind of talk specifically about the projects that I'm working on. So the gelatin production, we're actually just constructed our plasmid and are waiting to receive it to actually transform our rice cell lines. And in parallel, we're developing kind of the validation assays that we are going to need to verify the production of our gelatin. And then with that other project, where we're using the rice cells as microcarriers themselves, that's still in the kind of early literature review phase.
Yeah, and I'm in the process of designing the actual cultured meat facility. So yeah.
Awesome, cool. So are you guys working in collaboration because you're with some of the other labs here at UC Davis, since we have so many people working on this, or is it still kind of the early stages before that collaboration is really taking shape?
So far, it's still early, but we are very open to collaboration. Yeah, these are very new projects, but yeah, good question.
Yeah, we should talk offline about that.
Definitely. Yes, please.
Okay, awesome. Well, thank you everybody for joining us for the Academia Lab. Thank you, Patrick, Justin, Brian, and Kelly for all of the content on your research, and I'm stoked to see what you guys come up with in the future. So I'll definitely be keeping track of you.
Thank you,
and
Nick.
Thank you, ProteinX, for having us all here to share this.
Yeah, thank you, Nick. Thank you, everyone. It was really great to see new research and see what you guys are doing right now. So thank you very much.